Hi, Lily, can you hear me?
Yes.
Okay. Hello, everyone. My name's In. So I'm the product manager from CUSABIO. I'm so glad to see you in our webinar. Today, our topic will about the knowledge of ELISA high-throughput detection technique. And for today's webinar, we will talk two parts. So the first part will be a brief introduction of the ELISA principle and the different types of ELISA. And then for the second part, I will introduce multiple parameters of ELISA case. And I will show you the cases about our case, mainly about the performance comparison between our case and different vendors. So let's start the journey today. The first part is the ELISA principle. And ELISA, actually, the name is Enzyme-linked immunosorbent assay. This is a great technique to detect the presence of the antigens in biological samples. So basically, it relies on the interaction, especially the specific interaction between the antibody and the antigen. And the result usually shows in our enzyme to catalyze the change in the color fluorescent, and the signal that you got will be highly correlated to the amount of the analyzed present in the original samples. This technique has been developed for many years, and there are now countless variations of the test that people can use it. So, and also there are various ELISA-derived technologies that emerged, and they showed great potential in different applications. So for example, here is for like a derived technology from the sandwich ELISA. So this is basically based on a high affinity within biotin system. And because the high affinity that exists here in the system, people has been applied it into many protein and nucleic acid detections, and also the purification method. So the other applications, actually there are a lot. People may be familiar with the diagnostic test, like use the test kits to test the hormone, tumor markers, viral contaminants. And they are widely used in different areas, such as the immunology, biological pharmacy, diagnostic industries. So I will list some screening tests. So for example, the screening test for the HIV infection, and also the pregnancy test kits. They are all based on the ELISA test kits. And also some other available kits, such as the detection of dengue fever, the TB, and the viral infection. Also, people can use the kits for the detection of the different food allergens. So I talk a lot about antigens that you can be detected with the kits, but I think the same can happen with the antibodies. So for example, after the infection or during the drug discovery, the kits can be used to detect the presence of the antibodies and also the autoantibodies. So this can be found in some patients that with the immunodeficient disease. And also the kits can be divided into different main categories, like the quantitative one and the qualitative one. And also they are compatible with the different sample type. So like the body fluid samples, the blood sample, the plasma sample, and the serum samples. So like, I think all these applications, they are based on the advantage of ELISA technology. So overall, the ELISA technology is high sensitivity and is also high specificity. And also the kit is very easy to use. The assays can be easy to perform in a high throughput and cost-effective way. So there are a lot of advantages that I will not list all of them. And according to how the mechanism of the ELISA, they can be divided into 4 different major types. The direct, indirect, sandwich, and competitive. So I will explain them one by one. First, the direct ELISA. This one is a very straightforward. So based on the name, you can think about the direct interaction between the antigen and the antibody. So as I showed here, the antigen is immobilized on the solid surface. So for example, the 96-well plate, and then you can add the conjugated primary antibody to link with the antigen. So because the specific interaction between the antigen and the conjugated primary antibody, after the addition of the enzymes, the reaction will produce a visible marginal signal. So based on the signal, you can detect the analyte. And I say that is direct ELISA because here, there's no secondary antibody need to use here. But comparing to it, we have another type, the indirect ELISA. So with in the indirect ELISA, is actually a comparing to the previous one. This one is a two-step process, meaning that you need to have another conjugated secondary antibody to help to produce the signal. So the antigen is immobilized and the primary antibody is added to interact with antigen, and the conjugated secondary antibody is added. So with the addition of the substrate, the signal will produced. So this is very two basic ELISA type. And the next two will be more complex than these two. The third ELISA type I will say is the sandwich ELISA. For the sandwich ELISA, it's slightly different from the previous two and even the first one, because this one, they need to have two specific primary antibodies. These two specific antibody, they can recognize the same antigen. However, the recognition epitopes are different for these two primary antibody. Here, I have a very simple workflow for the sandwich ELISA. So the first, the primary antibody is coded on the well. Then we can add the antigen that we want to measure. So antigen is binding to the primary antibody. Then we will add another second primary antibody, and this primary antibody is conjugated. So then after the addition of the substrate, the signal will produce because the second primary antibody is conjugated. You will get the signal. So the idea here is that the antigen is not directly immobilized on the solid surface. It's immobilized with the help of the capture antibody. Then you will have the signal with these two primary antibody. Comparing to this one, the last one is slightly different. Instead of using the two primary antibody, you need to have two different antigens. So the antigen is target antigen and also the conjugated inhibitor antigen. So here the same, we have a general workflow here. The first, you have the excess primary antibody coded on the well. Then you can add the conjugated inhibitor antigen together with the target antigen. So there will be a competition between these two antigens. And so after the addition of the substrate, the signal that you get actually is from the inhibitor antigen reacting with the primary antibody. So then you can think the signal that you got actually is inversely associated with the antigen of interest. It's different from the previous two, like the more amount of the target antigen in the sample, the stronger the signal. Actually, for this competitive ELISA is different. The lower the amount of the target antigen in the sample, the stronger the signal, because the signal actually is coming from the inhibitor antigen interaction. So after I talk about the 4 different types of ELISA, hopefully you have a very basic idea about the ELISA principle. And to have a deep impressive about this, I make a short summary here about 4 different ELISA types. So, and all of them, they have a like advantage and a disadvantage. And the applications for each type of the ELISA can be slightly different. So for example, the direct ELISA, this one is pretty simple and quick. And because only one antibody used, so no signal amplification will happen with this type. But for the indirect ELISA, the signal can be amplified because we have the secondary antibody added. But because the secondary antibody is added, the non-specific signal and the cross-reactivity may be induced because the addition of the secondary antibody. But the advantage because of this is that this one is more high sensitivity and also is more flexible than the direct one. And also because the slightly difference of the application can be different. The direct ELISA is usually used to analyze the immune response, and the indirect ELISA can be used for detection of endogenous antibodies. And I guess this is because it's more high sensitivity. And then for the sandwich ELISA and the competitive ELISA, just be sure that for the sandwich ELISA, you have the 2 primary antibody here. And for the competitive ELISA, you have 2 different antigen, especially the inhibitor antigen. It's needed. So these 2, the protocol will be more complex than the previous two. And also the sandwich ELISA, because you have an additional primary antibody. So the price will be slightly higher for this one comparing to the other three. And also the applications for these two are also different. Like the sandwich ELISA, they are usually generally used to detect the macromolecules, because the macromolecules, they are large enough to bind to the different antibodies. But for the competitive ELISA, because you have the inhibitor energy and it's more higher sensitivity, so it's usually best for the detection of small antigens, even with very low concentrations. So this is demonstrating that it's pretty high sensitivity. So overall, I will say that they are very helpful. And I will say that based on what you need and based on your experiment, be sure to choose the appropriate ELISA to work with, because you see the complications, they are slightly different. So after I talk much about the ELISA principle, the next part, I will talk about the parameters and the performance from the CUSABIO ELISA kit, and also hopefully with the case that I displayed, you will know the advantage of CUSABIO ELISA kit. So this is a very complete list about the reagents for each CUSABIO ELISA kit that you will get. So like the assay plate, and you will get the standard protein, and also you will get different diluent and the wash buffer and the substrate stop solution. So each kit you will get like things like this. And the next, I will go through the parameters of on the product page about the CUSABIO ELISA kit. So first, the website of our CUSABIO is cusabio.com. And we have a search box on the official website. If you know the code or the catalog number for the CUSABIO ELISA kit, you can just simply type it on the search box. Or if you do not know about it, you can just simply type the abbreviations for the protein and the gene names in the search box. Then you can also enter into the product page. And on the product page, you will find the code. This is very convenient for your next time, like if you need to use the kit again for quick search. And also we have the abbreviations here. Just be sure that you remember it, because we'll make everything quick and convenient. The first parameter that I want to introduce is the detection range. So people care about detection range because this is the detectable concentration range. And that for your test, because you may want to know that, do I need to perform the dilutions for my samples? And this will be very helpful to help you to prepare your samples. And the detectable concentration range will only available to the quantitative ELISA kits. So just be sure about that. And also the concentration that we got for this, they are actually calculated and based on the standard curve from our standard protein. So people can use different softwares to make this standard curve and then to make the quick calculation about the concentration. And next, the parameter is the sensitivity. Sensitivity is the lowest measurable values that for the analyte, and is usually calculated based on the signal of the background and the inherent variability of the assay. So people care about sensitivity because they want to know that how low my sample concentration can be reached. So the lower the value, the higher the sensitivity. And also, in addition to this, I have a very brief comparison about the sensitivity between the CUSABIO and the other competitors. So the y-axis is the 21 targets that I make the summary, and the x-axis is the sensitive comparison. The blue bar is the CUSABIO ELISA kit, and the green and the red, they are from other vendors. So as I showed here, it's pretty clear that CUSABIO ELISA kits have a very excellent performance comparing to the other competitors. And the next parameter I want to introduce is the coefficient of variation. Let's just simply call it CV because it's easy. So CV is very helpful to identify other any inconsistencies and inaccuracy in the result. And it represents the precision of the kit and how many times that you can produce your data within and between the assays. So to confirm that we have a very good test about our kits, we did intra-assay precision test and the inter-assay precision test. So for these two sets, so intra-assay test, we have 3 samples that we have a known concentration. And then we test all of them 20 times on one plate to make the assessment. And for the inter-assay precision test, we have the 3 samples of known concentration, but we test them in 20 assays to X. So as I showed here, based on the test result we got, you see that for the intra-assay precision, the CV value, they are less than 8%. And for the inter-assay, the CV value is less than %. I guess that based on what I understand about ELISA kits, these 2 values are pretty excellent. So you want to have a low CV because you want to avoid the large variance that you got from your real test. The next 2 parameters I will talk together is the linearity and the percentage of the recovery. Because these 2 together, they are the key performance criteria to make sure that the assay is accurately detecting our samples and then making sure that the assay will be ideally for this sample matrix. So to confirm that, we also did different tests. So the first the sample matrix is serum, and then the other one we did is the cell culture supernatant. And also I want to emphasize that the linearity of the assay is pretty important because usually our samples, they sometimes have a very high concentration, and then you probably need to perform different dilutions. Then you want to like the actual concentration be the same as the calculated concentration after multiplying with the dilution factor. So you want them to be pretty close to each other for each dilution factor, right? So we did this by spike with the high concentration of the CUMA ADP in various metrics, and then we dilute it. Then we make it the calculation result. So this is our test result. See, as far as I know, usually if the range is between 80% to 120%, we will say the linearity is pretty good. And actually our linearity is pretty, falling to that range. So we have a very good performance or good parameters about the linearity. And the next parameter, as I mentioned, is compatible with the previous one. This one is the percentage of the recovery. This one is determined by the spiking a known amount of the purified target protein into the biological sample type. And we want the spike samples is measured with the ELISA, and the concentration is calculated from the standard curve. So you always want the calculated concentration comparing to the known concentration. So for example, if you have a 100% recovery, which means that the calculated or the observed concentration was exactly the same as the actual concentration of your spiked protein in the sample. So the higher, the better, right? And we did different tests here with different type, the serum sample type and then the ETTA plasma sample type. As you see, the average of the percentage recovery we got is 95% and 97%. And this is our range. So it still looks very good, right? So overall, based on our linearity and the percentage of recovery, we have a very good, excellent parameters with our ELISA kit. Then the next parameter I will talk about is the stabilities. So the stability is highly correlated that how long you can save your box and then how long they will be useful. And our promise is that after you get the box, after you get the kit, if they saved at 2 to 8 degrees, the storage time can be up to 6 months. And to guarantee this promise, we did different tests. So for example, we have a test that kids were placed at the 37 degree for 7 days, and we will compare the OD value, comparing to it the same product, which we placed at the 4 degree. And if the OD value is less than 30%, we will say that it's qualified. Then we know that our product is having a very good performance. And in addition to that, we also test with different temperatures, like 4 degree and 37 degree. And for the 37 degree, we even did a test with multiple days to make sure that our product is qualified. So overall, we have 3 different promises. Like we have the product inspected before the ship, and we guarantee that during the transportation, the kit is stable. So so do not need to worry about the stabilities. They are pretty good. And the next parameter is the specificity. So I guess some people may get confused about the specificity, but think about your sample. Sometimes the sample is not in a very optimal condition. Sometimes the samples, they are from the nature sample, right? So it should make sure that the kit can be specifically detected without cross-linking with other closely related or similar molecules. So let me give you an example. Because in some raw materials, there are so many substances there. So if we take this mouse TNF-alpha ELISA kit as example, for this the kit to make sure that only the mouse TNF-alpha substance can be recognized with this kit, but not other kits. We test the different substance. So we test the CD40, the TNF-beta, and then CD40 ligand from the mouse. And then we also test the same substance, the TNF-alpha from human and other species, just to make sure that only the mouse TNF-alpha can be identified and detected. Not all these substances that from other species or other molecules will be identified. So overall, this is to make sure that the target protein is coming, especially coming from the nature sample or the raw materials, will be selected with the high specificity. And this is to make sure that our ELISA kit is very, excellent. And in addition to this specificity, we also have the validations about the nature samples, because the kit not only, as I said, the kit not only perform well with the controlled condition, but also accurately and reliably detect the target analyte in the real complex biological samples. So based on that promise, we did the validation. So this is like some of the experiment data that we were provided with the kit to avoid, the contamination or the hemolysis with your real samples. So overall, I think after I introduce all the parameters, you will have a very better understanding about our CUSABIO kit. And in addition to that, from here, I will show you 2 different cases. So each case is a comparison between the CUSABIO ELISA kit with the other kits from the vendors. So basically just a performance comparison. The first case is the human interferon gamma ELISA kit. So this is an example plate layout from our comparison experiment. So first, we have the CUSABIO standard protein here, and we built the standard curve with different duplicate rating. So for example, the OD value number 1 and OD value number 2. Then we can, based on these 2 different OD, we will get the average rating and also standard deviation. From the average value and the standard deviation, we are able to calculate the CV. So as you see, the CV looks good, like all of them less than 8%. Also, we use the CUSABIO kit to test the standard protein from vendor A and the standard protein from vendor B. In addition to that, we also prepared 8 different serum samples and 8 plasma samples. And looks like that only the number 8 serum sample the CUSABIO kit are able to get the analyte measured with the exact concentration. So to be summary, for the CUSABIO kit, the CV is less than 8%, and the CUSABIO kit are able to detect the standard protein from vendor A and the standard protein from vendor B. And also for the samples that we prepared, only in one serum sample, we got concentration like approximately 210 picogram per ml. And for the other samples, we don't detect anything. So the next is, in this case, is using the vendor A vendor A ELISA kit and the vendor B ELISA kit to perform the similar experiment. And first, we have the standard protein from vendor A and the standard protein from vendor B. And we also built the standard curve to calculate the CV. So as you see, the CV from vendor A is less than %, and the CV from vendor B is also less than %. In addition to that, we definitely use the vendor A kit to detect the standard protein from CUSABIO and the standard protein from vendor B, and exactly the same. So we confirm that all three kits, they can recognize the standard protein from each other. And also, in addition to that, definitely we have the sample test. So list 8 serum sample and the list 8 plasma sample, they are the same. So we use vendor A kit and vendor B kit to perform the test. And actually the results are pretty similar. So as you see, the CUSABIO kit are able to detect the serum sample number 8, and the vendor A and the vendor B, they have the similar result. However, even the concentration are slightly different. As you see, the vendor A get the concentration like 65, and the vendor B get the concentration like the 96 picogram per ml. But overall, the trend, or I will say the qualitative signal, is exactly the same. So the case summary for this human interferon gamma ELISA kit is that all three kits that we got, no matter it's from CUSABIO or vendor A or vendor B, all three kits, they have a consistent higher sensitivity. But we found that the CV of CUSABIO ELISA kit is the smallest because, as I mentioned, CUSABIO have the CV less than 8%, but the other A and B, they both have less than 10%. So I will say that based on the comparison result we got, CUSABIO ELISA kit is more sensitive. The next case we take, for example, is the mouse interleukin-1 beta ELISA kit. So we performed a similar assay as the first case that I displayed, but for this time, the competitor we choose is the vendor C. So the results are pretty, so the experiment procedure pretty similar. We have the standard protein from CUSABIO and the standard protein from the vendor C. We also calculate the CV. The CV all looks better. Like, I think both of them, they are less than 10%. But in addition to the CV, we found that for the vendor C protein, the OD values, especially this low concentration group, looks like that the OD is pretty low. The value are very close to the blank wells. And I think that this will lead to some inaccurate measurement of some samples. So I think this is not that good. And we also performed a real sample test with CUSABIO ELISA kit and the vendor C ELISA kit. The samples that we prepared, they are from the serum sample for the wild-type mouse, and measuring the interleukin-1 beta. So as you see, for CUSABIO kit, the concentration that we got for the serum sample, the range is from non-detectable to 60 picogram per ml. But for the vendor C, the concentration range is pretty large. It's kind of like varying from 42 to 225 picogram per ml. And based on our research on the literature, I think the concentration range from CUSABIO kit is close to what have been reported in the literature. And comparing to the CUSABIO, vendor C, the concentration detected is definitely high, very significant higher than what have been reported before. So maybe not that good. And this is the case summary for the mouse interleukin-1 beta ELISA kit. So overall, both of the cases, they have a consistent higher sensitivity. We have the CV less than 10%. But I will say that the sensitivity for the CUSABIO ELISA kit is higher. And also for the real test with the serum sample, the test value we got with the CUSABIO kit is definitely closer to the reported value that we can find in the literature, which is very amazing test. Yeah. So I already introduced the parameter and the performance, and I want to show and also show you different cases about the CUSABIO kits. So by this, I want to summarize the advantage of CUSABIO kits here. So they are ready to use, and also our kits cover more than 30 species, including the human, mouse, rat, pig, sheep, goat, and monkey. And also based on the parameters that I introduced, we promise that the kits have a very good performance. They have a very good control to make sure that when we deliver to it, they will have a very good result. And also our kits are validated in multiple biological samples, including the serum, plasma, body fluid, and the cell culture supernatant. Also, in addition to that, we are very glad or proud to say that our kits have been cited in a lot of publications, like more than 20,000 reference. And for evidence, we have a partial list of the CUSABIO ELISA kit cited here. So more than 20,000 published paper. And like for the papers that I listed here, you may find familiar, Nature, Cell, Signaling, and Immunity. And here is the list of the CUSABIO products that have been cited in the paper and help these authors to get very amazing results and to facilitate the development of the research. At the end, please give me several minutes to introduce our company. So our company is CUSABIO. This is promised to our customers. So it means customer satisfied biology. And we have a very complete product line layout. So we do not only have the ELISA kit, but we also have the protein, antibody, molecular biology products, small molecular antigen. And beside that, we also have a very good recognition around the world. Our products has been exported to more than 80 countries, including the Europe and American, Japan, and then Germany. And also the cooperation between us and big companies, they are pretty excellent. We have a good cooperation with companies like the Pfizer, Bayer, Moderna, and Zenko, Johnson & Johnson. You know, all of this, they are very famous countries, famous companies around the world. So beside that, I will end this webinar, I will end this talk, and at the end, thank you so much for your listening, and we will be your good partner in the biology research. Thank you so much. And if you want to connect with us, this is the code for the LinkedIn, so please scan it and connect with us. Thank you so much.